The overall goal of the following experiment is to detect changes in hippocampal ERK phosphorylation following spatial learning in engrailed-2 knockout mice as a model of neurodevelopmental disorders. This is achieved by first testing both wild-type and engrailed-2 knockout mice in the Morris water maze. As a second step, the mice's brains are removed and slices from wild-type and knockout mice are prepared by vibrotome cutting. Next, brain slices from wild-type and knockout mice are processed for immunohistochemistry and used to detect ERK phosphorylation changes in the hippocampus. The results show differences in hippocampal ERK phosphorylation between wild-type and engrailed-2 knockout mice based on light microscopy visualization of the immunohistochemical staining.
This method allows to identify specific subsets of hippocampal neurons that are activated following a hippocampus-dependent spatial learning task, such as the Morris water maze.
We first had the idea for this method when we decided to investigate the molecular cascade that underlie the molecular deficit in engrailed-2 knockout mice, a mouse model for autism spectrum disorders.
To begin, train mice from various backgrounds of interest in a spatial learning task such as the Morris water maze. Follow the training regimen as described in the accompanying text protocol. At the end of the training period, euthanize the mice and then fix and remove their brains. When ready to continue with the procedure, use a scalpel to remove the cerebellum. Then superglue the rest of the brain upright onto the holder plate of the vibrotome.